I think you are muted.
Yeah. I hope am I audible?
Yes.
Am I audible?
Yes, you are.
Yeah. Glad to see you here, sir. Thanks once again for joining us. And it's so nice to see you. And good morning, everyone. And I hope we can start the session now. We are on live now. So I hope, Sneha, could you please start the session?
Yeah.
Yeah.
Okay.
Dear participants, one kind intimation is that the live link got shifted to the other link. So kindly come into this link, and the previous link will be deleted shortly. Yeah, Sneha, meanwhile, you can start the session and the participants will be joining soon.
Okay. Good morning, good afternoon, and good evening, everyone. Distinguished participants, ladies and gentlemen, I'm pleased to welcome you to the webinar on current and future agriculture with genome editing. In this virtual conference room, I see such amazing guests, dignitaries from around the world sharing some interest and enlightening one another with advancing in the field of agriculture. I'm sure we are also reaching out to many new practitioners, technologists, innovators, business professionals, and civil society representatives, fellow mates, and many more people. A warm welcome to you all. We all understand the importance of science, technology, and innovation in our day-to-day lives and the ways in which they are transforming the world. If this was not clear, it is blindly obvious now alongside the intricacies arising with the hour at hand. Science, technology, and innovation are indispensable for our response to and upliftment from the current and forthcoming scenarios. Equally important are deployment of scientific inventions, innovations, and technological solutions on a scale that will reach their target audience. Why here? Why today? And what is the purpose? We are all embraced by the presence of the distinguished and well-rounded dignitaries to shed some light upon the varied measures to be taken under consideration for the distinct genomes and the selection of crops based on the same. All our online webinars aim to discuss leading-edge technologies and recent developments in addition to the immediate challenges in agriculture and allied sectors. The idea is to familiarize students and professionals about current research trends, relevant and pioneering technologies in plant genomics for high-level analysis and interdisciplinary areas that are embraced in leading laboratories by the excellent scientists. Plant Genomia is a non-profit organization promoting and publicizing plant sciences and investigations in the field of agriculture across the globe. We organize various webinars and developments comprehending plant research. Here at Plant Genomia, we hold a global team of reputed scientists and research scholars from various parts of the world, distinct states of India, the United States of America, United Kingdom, Nigeria, Philippines, and Spain. Technical support is laid up by the scholars from the Department of Genetics and Plant Breeding. Our motto is to disseminate every bit of knowledge among all. We believe that no research is accomplished until broadcasted, and that it is an ongoing process through layers and lapses of time. I would like to extend my heartiest welcome to the senior dignitary, Dr. Kutubuddin Ali Mullah, Scientist, Biotechnology, Crop Improvement Division, National Rice Research Institute, the core subject specialist team, the core support team, our esteemed founder, Mr. Alamaru Krishna Chaitanya, and the lovely audience. We have the much admired founder of Plant Genomia, Mr. Alamaru Krishna Chaitanya, to address the audiences and guests present with us today, and to remark the initiative taken by the Plant Genomia and his team. Over to Mr. Alamaru.
Thanks, Neha. Am I audible and clear?
Yeah, you're audible and clear.
Fine. Good morning, afternoon, and good evening to everyone who are watching us here in this virtual gathering all over the world. Welcome you all once again on behalf of complete Plant Genomia and team. I'm delighted to say you that Plant Genomia received a good number of registrations from almost 90 plus countries across the globe. We had achieved a good record of statistics in our webinars. Almost 30,000 plus people watched our webinars from 98 plus countries, and it was so amazing to share with you. With an aim to promote plant science research, Plant Genomia is organizing the talks from international renowned scientists who are well known for their research skills. Also, very soon by the next month, we are about to launch our scientific forum for getting the guidance from scientists to the selected participants in writing scientific manuscripts, papers, books, chapters, proposals, and many more in various disciplines of agriculture. Plant Genomia is all about knowledge dissemination and would be a package of library of several such initiatives in the near future. We thank you all for the kind support and participation. I must truly thank all the scientists and speakers for the guidance and encouragement to do many more such activities. Today, we had a well-renowned speaker who is a talented scientist and an efficient researcher going to deliver a talk on current and future agriculture with genome editing. This topic is very crucial as there are a lot of challenges in agriculture, and genome editing is the solution for most of them and is a revolution of molecular sciences. The Royal Swedish Academy of Sciences has awarded the Nobel Prize in Chemistry 2020 to Emmanuel from Max Planck Unit of the Science of Pathogens, Berlin, and Jennifer from the University of California, USA. And I'm glad that today's speaker, Dr. Molla, has also associated with them while he was in abroad. I also heartfully put my sincere greetings to Director of NRRI, Cuttack, India, for nominating Dr. Molla as our speaker, who has in-depth insights into this subject of genome editing and CRISPR-Cas technologies. In this regard, our speaker has chosen such a booming topic to provide the insights on this ongoing and emerging scenario. I heartfully thank Dr. Molla for accepting our kind invitation in very less time out of his very busy schedule. Thank you.
Thank you very much. Those were some very gripping words coming from you. While we are presenting a gesture of gratitude, I would also like to thank Dr. K.P. Vishwanathan, former Vice Chancellor of Mahatma Phule Krishi Vidyapith, Rahuri, and Dr. K.V. Peter, former Vice Chancellor of Kerala Agriculture University, for giving their valuable forward message on Plant Genomia webinar. Before we begin, I would like to introduce Dr. Sujay Shri from IARI to introduce our revered speaker, Dr. Kutubuddin Ali Molla.
Thank you.
Welcome.
Kutubuddin Mullah is a scientist in agriculture biotechnology at ICR National Rice Research Institute, Cuttack, India. He has obtained his PhD from University of Calcutta. Dr. Mullah has done his postdoctoral research at the Pennsylvania State University, USA, availing the prestigious Fulbright Scholarship. He is interested in precise genome editing and uses CRISPR-Cas and other advanced editing techniques for crop improvement. Dr. Molla has been awarded the Young Scientist Award by the Indian Science Congress Association, Kolkata, Jawaharlal Nehru Outstanding PhD Thesis Award by ICR New Delhi, and INSA Medal for Young Scientist 2020 by the Indian National Sciences Academy, New Delhi. He is also the recipient of a AAAS Science Program for the Excellence in Science from American Association for the Advancement of Science, Washington, D.C. Dr. Molla recently got selected as a fellow member of Indian National Young Academy of Sciences, New Delhi. Dr. Molla regularly publishes his research findings in peer-reviewed, reputed international journals. He has co-edited 2 volumes of Springer book on CRISPR-Cas methods. He is currently serving as the editorial board member of PLOS ONE and Frontiers in Genome Editing journals. Sir, thank you for accepting our invitation to be a part of Plant Genomia. Now I request you to share your screen and start the presentation.
Okay, wonderful. Thank you very much for your wonderful introduction and wonderful introduction to this initiated by Plant Genomia. So it's good to see you that all of you are so young and started, I would say, as a kind of very bold step to communicate science to the general audience, also to the student researchers and whoever need to know the science. So it's my real pleasure to be here, although it is Sunday, but I enjoy talking on genome editing. Yes, I will be talking a little bit about current and future agriculture and how genome editing could impact these two current and future agriculture. So let me go to the slide mode. Yeah. Okay. So the answer you will get from my presentation today of these following questions: how we learned agriculture and became self-sufficient in food production as mankind. And the second question you would get answered from me is, what are the problems that today's agriculture face? And the third question I want to address here, that can genome editing play a role in solving those problems, and how it could impact current agriculture and future agriculture. So can anybody identify what picture it is that I am showing here to? Okay, let me tell you. This is a very old plant ancestor, wild ancestor. It's named as teosinte. And we took 9,000 years to develop into a crop out of this teosinte. And this process is called crop domestication. So, you know, in 10,000 years ago, we did not have crop like we have today. So we had only this kind of plants that was very few in grain number and was very difficult to grow in a compact space. So we modified them according to our need, according to our cultivation need, according to our taste need, according to our to accommodate many plants in a small space. All this kind of need we fulfilled by artificially selecting them. So now you can see here, the left panel is a white teosinte. You can see there, one single teosinte plant takes a lot of space and they go sparsely here and there. But what we modified to today's agriculture is one single maize plant is very stout and takes very small space. And they can be grown, a huge number of plants can be grown in a small space, which is very suitable for our artificial cultivation. So remember, agriculture never been a natural process. It is an artificial process that we developed for our own need. So we have artificially selected plants to increase their suitability to human requirements. Like according to our taste, we modified them according to the need of growing more yield from a single plant, and then storage, how are they suitable for long-term storage or not, and then also for according to our cultivation practices. You know, if I want to give you some more examples, so here you can say this is a wonderful slide. I think a lot of you have already seen it. So this is a common wild master, and when we deliberately selected for a desirable trait like stem, then we developed kohlrabi. When we selected for lateral buds, then we developed Brussels sprouts, and similar, when we go for terminal bud selection, then we develop cabbage and like cauliflower, broccoli, and kale according to our need. And you can see that these all vegetables that we eat today, these are all because of artificial selection. They never developed naturally. Okay, so in 1800s, mid-1800s, Gregor Johann Mendel, who is the father of genetics, then he outlined the principles of heredity, and that was the starting point of scientific plant breeding. So we know how a trait inherited from one generation to another, and what ratio they are segregated out. So that helped a lot to the plant breeders to incorporate a desirable plant trait to a cultivated varieties and tracking them to develop a improved variety. Now, if I want to show you some kind of genetic variation, so this is a single species Urania sativa, and you can see a huge type of phenotypic variation in their seeds only. I'm not showing you the plant architecture and all this. This is only the variation in seeds, and this photo was taken by one of the smart students from NRI, right, Pritesh. So why this kind of variation exists in the phenotype? So this is all because the spontaneous mutation that nature do. It is not no one artificially induced those mutations. Mutation means that DNA modification, some kind of changes in DNA is called mutation. So in nature, they changes DNA for the benefit of the plants, and those change is called spontaneous mutation. Okay, so up until 20th century, we did not have any other means other than a spontaneous mutation to exploit in selecting plant and animals. Suppose, you know, suppose I like the top one. One second, pointer. Okay. So suppose I like this one, someone could like this one, someone could like this one. Okay, so these are all our specific needs. But suppose in Tamil Nadu, in Andhra Pradesh, some different kind of likings. In West Bengal, Odisha, we have some different kind of likings. So you have to develop varieties following that likings. Okay, so the only options to breeders was to utilize those variation was generated by spontaneous mutations that is already existing in nature. So natural variation. Now, when we learn that genetic variation is the key to crop improvement, because phenotypic variation and desirable trait all develops from genetic variation. When we learned that, then we pondered how to artificially generate genetic variation, as we cannot wait for the spontaneous mutation to happen because we have to feed our growing population. So we always try to artificially generate genetic variation. Say 1920s and 40s, at that time was pioneering work has been done to induce artificial changes in DNA by applying physical mutagens like X-rays and gamma rays, and also some chemicals like alkylating agents like EMAs, ethyl methyl sulfate, and all those things. So you can say this is an example of mutation breeding. So this is the original flower. And now when it was, the seed of this plant was treated with gamma rays, and it developed, the progeny developed a lot of different kind of colors due to the random mutation generated by the treatment of this kind of gamma rays, which induces changes in DNA. But remember, this kind of mutating agents or mutagens, they create random modification or random changes in DNA. You cannot control which spot you want to modify, which spot you want to change. Okay, so this is kind of random, and then you have to grow the whole mutant population, and then you have to go for seeing whether you get a desired trait or not. So it is all by chance. You can get a desired trait that you are looking for, you may not get also. Now, you know, according to a Food and Agriculture Organization statistics recently, that more than 2,500 plant varieties developed by radiation-induced DNA modification till now. So now, but now today's agriculture and future agriculture is expected to suffer from numerous problems. Here are some of the, from this infographic, I want to depict you some of the problem, as we have, you know, growing population that is expected to reach 9.8 billion by 2050. And then we have reduced groundwater content, so we fight for water. So, you know, agriculture also needs water. So how we would be able to supply water for our agriculture? Then we have a shrinking agricultural land and arable land, so we have problem to grow more food in a limited space. And this is another very, very emerging problem that we have to think about, that is climate change. So, you know, agriculture is hugely dependent on climate and weather. So in a changing climatic scenario, agriculture would be impacted severely. And as the temperature grows, goes down, then the pest and pathogen dynamics also changes. So new pest coming and the old pest going, new diseases coming, emerging. So farmers are facing a lot of different kind of new problems they have never encountered. So we have to develop such crops that could withstand pest and disease, and that have a stabilized and increased yields despite of changing climates. And also we should develop crops with minimized resource input, suppose very few amount of water, very less amount of fertilizer is also sufficient to grow that kind of crop. So we have to go for that kind of. So how, now the question is how we could go for developing those crops. So we have to go for precise breeding. So as I told you, the mutation breeding helped us to go beyond the spontaneous natural mutation. We could learn how to artificially induce mutation, how to artificially change DNA letters. But what if we have tools or methods that allow us to modify DNA at a desired spot that you want? It's not random, because random takes a lot of time to select, to purify. But if you have a tool that could modify your DNA in a location that you want for, so I call it precise genetic variation on demand. So I'll be talking about a lot about this one. So before jumping into those kind of precise changes in DNA, you have to grasp this idea of what are the building materials of a human. A human is made up of 37 trillion of cells. Similarly, a higher plant like maize, rice, they're also made up of trillion of cells. And this is an example of plant cell, and you can see this nucleus is very important for this cell because it contains chromosome. And this chromosome is nothing but a coil of DNA. It's a long thread of DNA that coiled around histone protein to form this chromosome. So, but the long thread of DNA are not equally important. Some portion of that DNA is important because they are functional, and that functional portion of DNA is called the gene. Now, what is there in gene and why it is functional? Because when this gene expresses by forming mRNA and then protein, it expresses directly or indirectly a character, or it controls a character. That's why gene is called a functional unit. So if you see in the molecular details of that gene, what is there? So our DNA or gene does not have anything else other than this simple explanation that it has 4 different kind of DNA letters. One is adenine, one is cytosine, and thymine, A, G, and CT. And the sequence of these 4 letters actually makes a gene different from other. So gene 1 has a different sequence of these 4 letters. Gene 2 has a different sequence of these 4 letters. So, as I introduced you to that, do we have a tool to precisely modify DNA in a targeted location? So this tool that I'm going to describe in my whole presentation, that's Crispr Casting, is an amazing tool that can cause a cut in a desired DNA location. So it can make a precise double-strand break in the DNA location that you want for. So you could call it as a scissor. So if I want to give you an analogy how it works, so CRISPR Cas9 is a 2-component system. One is a customizable RNA, and that we call it as a guide RNA. Another is a Cas9 protein, that's a DNA cutting protein. So if I want to give an analogy, this RNA is an address, is a genomic address, and this DNA cutting protein or Cas9 is I call it as a postman that has 2 scissors in its pocket. Now, when you give the address to the postman takes the address in its pocket, and it goes to the genome that you are looking for modification, and then it searches for the address that you gave in the form of RNA. Okay, whenever it finds similar nucleotide or similar letter as it is present in the RNA, whenever it matches the sequence in the DNA, then it will bind there like this, and then the postman gets the signal that this is the region or location where I need to make a cut. Then it uses its 2 scissors to cut the 2 strands of DNA. As you know, that DNA is a double-strand structure. So here it makes a double-strand break. So if it is a gene or functional unit, if you cut it, then it becomes non-functional. Now, as I told you, CRISPR-Cas9 can make a cut. It is clear to you, I guess. But how it causes mutation? The answer is here. You understand that double-strand break can be made in a desired location as you want. Whenever there is a double-strand break, cell shake itself because it is very serious damage to cell. Because DNA is the hereditary material, it's a genetic material. So the damage to DNA means the code for running the cell gets damaged. So that's why it needs to repair that. So cell immediately deploy its cellular repair mechanism, and one of the predominant pathways is non-homologous end joining that repair the cut to seal the two broken ends. But this one, repeated cutting and repeated repairing sometimes causes some error to occur. And due to some erroneous repair, this pathway could also randomly insert or delete some original bases or insert some new DNA later in the cut part that I have shown here in the red color. So, as I told you, gene 1 is different from gene 2 because of the sequence. Later is same. So whenever there is a new random insertion or deletion of DNA later, then the sequence gets disturbed. And as the sequence gets disturbed, the open reading frame gets disturbed. And then it cannot produce the same kind of protein as it was producing in its original form. So the gene gets knocked out. So it means gene gets non-functional. So you can generate very easily a mutant of a gene in order to see whether this gene functions for this purpose or not. So it is a very efficient tool to generate knockout mutant. So this video will allow you to understand in more detail.
Been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, good. So this, this brown color base pair, this new base that was inserted in this example causes the disruption of the open reading frame so that they cannot produce the protein anymore. Okay. As I introduced this lecture in the beginning, I told you about domestication, and it takes 9,000 years to domesticate from Teosinte to modern day maize. So I want to give you some example of how genome editing is actually helping rapid domestication. So, this is the vorager sativa that we eat, the rice plant. This is a domesticated diploid, and this is an vorager alta that is a wild ancestor. It's like Teosinte. You can see the growing pattern of this plant is so vigorous and it takes a lot of space. And also this is a panicking. This does not look like the cultivated one, so it needs a lot of modification to make it a cultivated one and domesticated one. Okay, so recently genome editing has been applied to domesticate this Orangea alta. It's like a domesticated crop to modify it. So you can see the problem of this Orangea alta is that many wild rice has this long on, because this is one single rice and this has a long on present, and this is a very negative character for cultivation, because you cannot, for threshing and all the other things, for harvesting is very difficult. And this is So when they edited, this is one single example I'm just showing you. When they edited a gene AN1, so you can see the reduction in on length, hugely reduced the on length, very small size compared to the wild one. So this gene AN1 is called a domesticated gene. So if you edit this domesticated gene, you can go for rapid domestication of wild ancestors or wild relatives, and it looks like the cultivated one. Okay, so there is another example I give you from a wild tomato. So this is a wild tomato, Solanum pimpanifolia menson. This wild tomato, you can see the growth pattern is so vigorous and it takes a lot of space, and it's not a compact one. So when the SP gene was edited, so this one is the edited one, and you can see the same plant grows in a very small space, and it does not waste space, so it is very compact in nature because of a single gene editing. And you can see also another problem is many wild species does not have a good kind of flower, a good number of flow rates, and it does not give you a good yield. So here is the wild tomato inflorescence that has maybe 5 or 6 flowers in one single inflorescence. When the multiflorate gene was edited, so you can see this is the edited one, and you can see how the inflorescence bounced, and it has, I think, 5 times more flow rates in a single inflorescence. So this is amazing. So you can see it takes only 2 to 3 hours to make a wild tomato look like a cultivated tomato. So how genome editing could reduce the 9,000 years into 10 years or maybe 5 years. Okay, so I'm not going into details of giving you huge example because all of you read a lot. So it has huge impact on the yield. So all major crops and minor crops are being modified for increasing the yield despite the climate change, and also disease resistance. There are huge examples of bacterial disease resistance, fungal, viral, nematode, and others also. And then it also playing a role in climate adaptation as a maize plant developed for drought tolerance, and rice plant developed for salinity tolerance, rice plant developed for flood tolerance with genome editing. There are huge examples, but I'm just giving a few examples. It can also impact on enhancing taste. So some genes that we domesticated our crop are not. We also breed our crop to look for a good marketing, to increase yield. That is called yield-oriented breeding. But we forget about taste, because you always complain to your grandmother or maybe grandfather that, when I was a child, this was very tasty. Now it does not taste. So it is because of our breeding. Breeder never cared about taste. Now CRISPR is helping to bring back those old taste in different kind of vegetables and also fruits. And also it has a huge impact in enhancing nutritional quality. As I can give you an example of healthy oil. So there is a soybean plant that has been developed that the oil composition is like olive oil. So it is very healthy, and this is already demonstrated, already commercialized in the USA. So wheat has been developed to remove the gluten allergy, to remove the gluten. Tomato with high lycopene. So these are all, you can play with the nutritional quality for the benefit of consumer. And then storage also. So you can increase the shelf life of tomato and also mushroom. So non-browning mushroom you always heard of probably. So these are all different kind of applications. You can Google it and you would find innumerable examples of this kind of applications. So also another point I want to give you an introduction to, that you could develop some new traits, brand new traits you never thought of, even plant breeder never thought of that trait. So this is an example, the rapid customization of tomato plants for urban agriculture. So you can grow it in your terrace, you can grow it in your balcony, because the terrace garden is mostly successful with lettuce and some kind of leafy vegetables, but not with any fruits. So this is an example how it could impact to develop new traits that is a need of present day human civilization. Okay, so I'm not going to give you further details, but it has been applied to almost all crops. So this technology is so easy to use, so available, and so easy to replicate that everyone is using this technology. Okay, so because of its huge impact, not only in agriculture, but also in human therapeutics and the basic biology that we investigate day to day in our laboratory, it just revolutionized the biological field, biotechnological field, and human medicine field. And that is why, recognizing the impact of this technology, within only 8 years, this technology has been awarded with Nobel Prize, and as mentioned by Alamu in the introduction. So it took only 8 years to get a Nobel Prize from the first publication to Nobel. Okay, so I give a broad example of the conventional CRISPR Cas9. Now I will take you to a different dimension to be more precise. So I brought you from phenotypic selection, depending on a spontaneous mutation, spontaneous DNA change, and then artificial DNA change, random DNA change by mutation breeding. And then I introduced you to the CRISPR Cas9 that could change or change a desired location. But have you thought that, as I described you, that CRISPR Cas9 can make a cut in the DNA or in a gene that you want, and then it inserts random insertion or deletion of some DNA letters, and that changes the sequence. So that is a very good tool to disrupt a gene sequence or maybe modify a promoter sequence, but that is random. What kind of insertion or deletion is happening, you cannot control. So what if you have a tool that would allow you to control what kind of insertion you want, what kind of replacement you want? So that I'm going to talk about. So, you know, people say that Cas9 creates random and unpredictable mutations in the cut point during the repair. But during our experiment, we saw a repeated occurrence of similar kind of modification when we use the same guide or when we used the same genomic address. So then we thought and we pondered that why it is happening. Does it mean that it is not random? And then we opined that it is not random and it could be predicted. So how it could be predicted? You know, a lot of people are using CRISPR nowadays. So if we have a big repair data means mutation data. So this is the CRISPR generated mutation. This is the base pair insertion. This is the base pair deletion that happens after this experiment. So if we have a lot of data, and then we can develop some machine learning model. And then after we develop this model, so this model could be able to tell you which guide RNA or which genomic address would give you good kind of mutation or good amount of mutation. And also it will allow you what kind of insertion or deletion would happen if you make a cut in this position. So let me give you an example. So if you see here, this is the target genomic region. This is the genomic address, suppose, and it will match with your genomic DNA. Now, if the -4th position from the PAM sequence is a T, then there is a high chance of you get another T inserted. If you have an A at the same position, you get high chance of another A inserted. If you have a C, if you have double C in that position, here, if you have double C in that position, you have high chance of one C deleted. So this opinion article has been published in CRISPR Trends in Biotech, and we are going to publish soon our finding on this kind of prediction. So yes, this slide that I showed you, it gives you some control. So if you have a predictor model in your computer and you put the genomic sequence that you want to cut, and then you can predict that it will create single base pair insertion or double base pair insertion or maybe 2 base pair deletion. So this would obviously allow you to somehow foresee your outcome, and foreseeing that outcome, you can design or changes the genomic address. Okay, so it would give you some kind of preciseness, but not an ultimate preciseness. Now I will take you to this direction. So this is a rice plant, or this is a Kitake plant that is susceptible, that is disease susceptible. It does not have the ability to resist a pathogen, but this one could resist a pathogen. But how? The difference between these two panel of plants is only a single nucleotide or single DNA letter. This susceptible one has C and this one has a T. So we know that C to T changes causes a plant from susceptible to tolerant. So our question is there a way to rewrite genetic letters specifically? So you have to be very specific to make a C to a T. So the answer is yes. This is the technology-based editing I will be talking about in my later part of presentation. So it works as a pencil, and it can be applied to change A to G or vice versa, T to C or C to T or C to G. So these are the tools that we have in our hand. So it is a modification of CRISPR-Cas9. Here this is the Cas9 that we call as N-Cas9, because this is a modified Cas9 that has a single caesar instead of a double caesar. And we fused a protein that is called deaminase protein with the N-Cas9. And this deaminase actually causes the rewriting of DNA letters. So here that I already described, it is called Cas9 or N-Cas9 that cause single strand break. Now, this video.
Those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay. So the first kind of base editor that has been developed from David Liu's lab, that is cytosine base editor that could cause C to T. Or in the opposite stand, it is G to A. So this is the architecture. I'm not going to go into detail of this architecture. There's another base editor called adenine base editor. There's a different version of base editor that could cause A to G, adenine to guanine. And those base editing field actually pioneered by this David Liu from MIT, very young, talented scientist, professor from MIT Harvard. So, this is the latest base editor that we got from 3 different groups that could cause transverse mutation. It's C to G. It could cause C to G. So if you have a DNA here that has a CG base pair, you could apply this CGBE base editor to modify it to a GC base pair. So CG to GC base pair, you could modify in your DNA with this base editor, and it is the first kind of transversion base editor. Now, I would show some of my brief example of my work with adenine base editor. So I want to highlight here this mechanism, how this adenine base editor work actually. Suppose you have a gene which has a defective base pair or nucleotide pair, that is AT. Now you want to modify this AT to a GC base pair. So how you do with the adenine base editor? Once you apply the adenine base editor here, then you can see this is the TADE. This TADE is called adenine deaminase. So this portion, they will deaminate the adenine and they will make it as a inosine. This inosine is a deaminated version of adenine. Okay. Now, so you can say this is the pair IT base pair. So it was AT base pair, now it becomes IT. Now this IT base pair is kind of, it is a mismatch, it's not a true match, it is a mismatch. So as I told you, N-Cas9 has a single scissor. It can make a single strand break, okay? So when there is an IT base pair in the genome, so the genomic repair mechanism, they also sense this kind of mismatch, and they say that, yes, so we have a mismatch here, we have to remove any of them. So which one is the mismatch? Is it the I mismatch or it is the T mismatch? So in order to trick the cellular repair machinery, this Cas9 would make a cut in this T containing strand so that cell understand that this strand has a damage, so T must be the wrong letter. Then they would remove the T from this strand and they will insert the right base, that is C, opposite to an I. So it become IC base pair. In the next round of replication, it will become a GC base pair. So you get your corrected gene by applying base editor in this mechanism. Now we try to demonstrate it in rice. So this is a gene was called white stripe leaf gene, and we targeted this A. We try to change this A to a G. Okay. So and another gene we also targeted. This is called Zebra3 gene, where we targeted this A. So we want to modify this A in the gene sequence. So this is a DNA double strand, how it looks like in a sequence form. Okay. So we targeted this one together, and we designed a smart designing where we means in a single experiment we could target both of the gene. So what we did, the address is here. This is the genomic address. This is another genomic address. This genomic address for the first gene WSL5. This genomic address is the second gene for Z3. Now they are fused with the tRNA. So when you insert into the cell, then the tRNA would be spliced out automatically by endogenous these two enzymes, RNase P and RNase Z. And then WSL5 is separated from Z3, and WSL5 would go to chromosome 4 because the gene is present in chromosome 4. Similarly, Z3 would go to chromosome 3 because the sequence matches with chromosome 3. Now, when they go there, base editor would also follow them. Now we tried in protoplast first to get an indication, and these, resistant band, RE resistant band is restriction digestion resistant band, actually an indication of successful modification of that targeting. So, this result indicated that we get successful base editing in protoplast. Now we moved forward to try it different kind of vectors into a regenerated plant. So we regenerated rice plants, and then we have seen in the first generation, in T0 generation, we have seen monoallelic editing. What is monoallelic editing? I will tell you soon. So here you can see this is the wild type that is not treated, and the Sanger sequence chromatogram is look like that. So if it is a green peak, it is an A. It is a black, it's G. If it is blue, it is C. And if it is red, it is a T DNA letter. So we have seen in our mutant plant that we got A changed to G, but you can see here two peak. One is green, one is black. It means the existence of both A and G. So rice is a diploid plant, so it has the same chromosome twice. So that's why it's called the same allele twice. So same gene has two alleles. Okay. So if one allele is edited, so it would look like this. If both the allele is edited, so it would completely convert into a black peak. Similarly, for the next gene Z3 also, we have found a very good monoallelic editing in our T0 plant. So it means we can modify both the location of DNA very specifically, the target DNA letter into A to G. Now, when we have grown them for next generation, we have seen a complete change, and both biallelic editing. You can see here, this is the control one, and this is the mutant. You can see a complete conversion of a green peak to a black peak, means A to G, by the application of this base editor. And when it is completely converted, we have seen the expected phenotype, WSL5 white stripe lived here. So this single letter change in the DNA causes this phenotype, which was induced artificially by base editor. Similarly, for Z3 also, we converted A to G, and we have seen this Zebra3 phenotype, light green, dark green, light green, dark green phenotype. And within a few months, in 4 months, we got base editor free, a non-transgenic mutant. So, the absence of band here, PCR band indicates the absence of any foreign DNA, but they are mutated. So how we develop? So, suppose when you transform your plant with base editor construct or base editor DNA, then suppose it is integrated into chromosome 1, but your target is in chromosome 4. Now, this base editor will produce a protein and it would go and modify the target. So once the target is modified, you don't need the base editor to exist. So if you grow the plant for next generation, it would be segregated out, and you have to select only the targeted mutant plants, not the base editor. So you can select those plants that does not have the base editor. So we can get a non-transgenic mutant very rapidly. So this is an exciting example that we achieved. This TAG, if it is present in a DNA, this actually works as a stop signal for the translation machinery, for protein synthesizing machinery. As we stop our vehicle to a red signal, so protein synthesizing machinery, they stop here whenever they see TAG sequence. So because of this TAG sequence always present at the end of a gene, so that the complete protein is produced and then it is stopped. But in some cases, some spontaneous mutation, some rice plant are in some gene, these stop signal comes at the beginning or at the middle of the gene, so that the total protein does not produce because of this mutation, and the plant becomes disease susceptible. So we tried, can we go for modifying this? So we tried with base editor and we modified TAG to TGG, and we get full length mutant plants, because when TAG modified to TGG, it is no longer a stop signal. It codes for tryptophan mutant, tryptophan amino acid. So the whole long protein is produced and the plant becomes disease resistant. Okay, so recently in September 3rd, just last week, this week, September 30th, we published this one. This is a very comprehensive and concise article on this precise base editing in plants. So, this is I never talked about before any of my talks. So this is organelle-based editor recently developed, pioneered by same David Liu, and it has been rapidly adopted for plant also. So, nucleus, I talked about nuclear DNA variation and all these things, but plant cell has also mitochondria and chloroplast. That has also DNA, that has also genetic material. That is also important for crop breeding, for our crop improvement. Because, mitochondrial gene is very important for hybrid crop development, because these mitochondrial genetic elements control the male sterility part of that hybrid compound. If you want to go for hybrid rice development and another hybrid crop development, you have to depend on the genetic variation that is present in the mitochondrial DNA. So it is very important for our crop. So another is chloroplast DNA. So chloroplast DNA is also very important for our yield because it controls our photosynthesis. And Rubisco, some part of Rubisco enzyme is coded from the chloroplast. So it directly impacts the yield of plant. So that genetic material also is very important. And if we could generate the variation of mitochondrial and chloroplast DNA, it would also help us to cope up with the climate change, cope up with the changing scenario. So this organelle-based editor allow us to change DNA in mitochondria and chloroplast. So I would recommend you, I'm not going into detail of the mechanism, but I would recommend you to go through our review articles to understand how it actually works. So you can change both cpDNA and mtDNA by this kind of base editor. Okay, this is the last technology that I'm going to share with you, that is called prime editing. And if I call CRISPR Cas9 as a scissor, base editor as a pencil, or you can erase one wrong letter and introduce the right letter, prime editing is like a word processor. So if you have a chunk of DNA letter that you want to replace with the correct letter, then you have to use this prime editing technology that has been recently developed in the same lab, WU, by a very intelligent postdoc, David Angelon. So this is a modification where Cas9 is fused with the reverse transcriptase enzyme, and it allows us to perform all 12 different kind of base substitution. You can insert precisely up to 44 base pair that has been demonstrated, and then you could delete up to 80 base pair of original sequence precisely, and then combination of above modification also could be done with prime editing. So you could also consult our review article to know more about this technology. So this is the last few slides in my presentation that I would give you a glimpse of how B's and P's, base editors and prime editors, is impacting crop improvement. I'm not telling you anything about how these two technology actually could impact in basic biology, but we discussed basic biology in our review article recently published in that Nature Plan. So you could go and different kind of genetic modification we could achieve with these two technology, future modification we could apply for. So you could consult that one, but I will discuss here only the crop improvement application that has been demonstrated already. Although it has a tremendous potential to apply it in many fields, but these are only demonstrated examples that I have shown here. So, one very good example is herbicide tolerance. Weed is very, very problematic for today's agriculture. If you want to grow our plant in less water, in aerobic condition, in direct seeded condition, then you have to go for some kind of herbicide tolerant plants. But how you develop herbicide tolerant plant? So for weed management, herbicide is one of the most efficient method. So how this actually mechanism works? So this is a very crucial enzyme in plant that works to develop to biosynthesis amino acids, and that actually helps in surviving the plants. Now, when you apply herbicide, this herbicide has a property to modify the enzyme or to block the binding site of that enzyme, that crucial enzyme. So if the enzyme active site is already occupied with the herbicide chemical that you spray, then the substrate will not be able to bind. So the enzyme cannot synthesize the amino acid that is needed for the plant. Now, if it happens and the weed dies when you apply herbicide, this is the mechanism behind it. Because the weed can no longer synthesize the amino acid for its own growth. So it dies within 2 days. Now, what if you spray herbicide, only weed dies, but your plant survives, your crop plant survives. So your crop plant has to have inherent ability to tolerate the weed. How? So suppose the same enzyme is present in your crop plant also, and now you modify single amino acid of that protein so that the binding site is modified. The enzyme's binding sites is changed as you changed an amino acid by genetic modification. Then the herbicide would no longer be able to bind to the enzyme of your crop plant, as it is already modified. So herbicide cannot bind and substrate can bind, so the enzyme can continue to produce the amino acid required for plant survival. So your modified crop plant can tolerate herbicide. Whether the weed, those are not modified, they will die. So this has already been applied in a lot of Arabidopsis paper, and these are the crops that I just showed you here. These are all has been modified with base editors and prime editors to tolerate different kind of chemical herbicides, not a single herbicide, but different kind of chemical herbicides. Okay, so this base editor has been applied to alter starch composition of rice. It means you can increase amylopectin, you can increase amylose as per your need, and this has already been demonstrated. And also in the strawberry, the sugar content was increased by application of this base editor. Tomato has increased carotenoids by the application of base editor. Similarly, the paper that I showed you, Orija Alta paper in Sale recently published, they have applied base editing technology to rapidly domesticated one rice plants. Okay, so next, very important portion that also related to climate change and new emerging disease. So if you want to develop a sturdy disease-resistant rice plant, so suppose we know from our natural biological experiment that a susceptible allele and a resistant allele has a difference on few nucleotides, and that few nucleotide could be changed, as I showed you by own example, stop codon to a tryptophan codon, from a susceptible allele to a resistant allele. So you can mimic a natural variation by applying base editor to develop disease resistance. And also this has been hugely being implicated in yield improvement. So these are the three that we highlighted. Stop codon in negative regulated genes, those genes that negatively impact in rice, negatively impact in yield, you could generate an artificial stop codon at the beginning or at the middle by using base editor, so that the gene becomes disrupted, non-functional, and your negative impact is gone, so yield is increased. Similarly, for haploid induction, so double haploid generation, haploid induction is also very important for breeding. That could be done, and also mimicking natural allele for high yield. Input use efficiency also we need, because we need low input use efficiency, because if you apply more fertilizer, it also emits more greenhouse gas. So if you apply less fertilizer, it is good for our environment, our soil health and water health. So there are genetic variations that actually increases the uptake, transport, and responsive useness efficiency of a plant. So that kind of genetic variation it could artificially generate P and P in a very less time. So I'm at the end of my presentation. If you want to further read about this exciting technology and want to apply in your experiment, in your project, future project, PhD project, or any kind of experiment, you could go for these. We published 2 volumes, CRISPR-Cas methods. So it actually it jotted down all the protocols to apply this exciting technology to your own crop, and it actually intended to give a ready resources, a handbook to a researcher who are new in the field. So this 2 volume could be consulted. Total maybe around 50 protocols you could get from these 2 books. And if you want to know more details about the technology, how it works, so the recent review, as I told you, in Nature Plant, and this one from Trained in Biotechnology, also consult. This is my group. I closely work with Dr. M.G. Beck's lab and collaborate, and we utilize the funding from ICAR, mainly NRI, National Science Fund, USA, USD, and Fulbright also. So this is to acknowledge them. Thank you very much, and I would love to go for further discussion if it is needed.
Thank you very much, sir. I hope I'm audible.
Yes.
Yeah.
So should I stop sharing?
Yeah, yes, sir.
Okay. Thanks.
Yeah, we are having some couple of questions for your interesting talk. So I would like to present them here. So the first question is by Dr. Vidya. What are your views on revolutionizing the future agriculture with genome editing?
This question probably she asked it during the initial part of my presentation, because I already talked about the agricultural field, how it is being revolutionized with genome editing. The main thing is that, a genetic modification that we used to do earlier, the transgenesis, means you see, transgenesis or transgenic generation has an added advantage that you can use the whole gene pool, not restricted to rice or wheat. Suppose you want to modify rice, okay? Now, some gene you find very useful from a mango plant. Now, you cannot transfer the mango gene to a rice until and unless you use transgenesis. So if you use transgenic technology, then you can take the mango gene out and then into the rice plant, okay? So you get to use the beneficial impact of a mango gene. Now, for genome editing, you are modifying the own gene, rice own gene. So you are creating variation by the existing gene pool, okay? But if you want to go for an out-of-box trade, then you need genetic modification using transgenesis. Another main advantage of genome editing, why people are very interested, because a lot of people, they have a negative feeling about genetic modification using transgenesis. But this genome editing technology allows you to develop a mutant plant, to develop a modified plant, but there is no foreign DNA inserted. You can segregate out. Suppose you apply Crispr Cas9. So once the Crispr Cas9 makes a cut and modify the bases, then you don't need the Crispr Cas9 to be present in the cell. So you can just get rid of them and you select only the modified plant. So your plant, modification done, but there is no foreign DNA. So that's the main thing. I think it seems too much attractive to the people. And that's why they are very much interested and eager to use it, solving a lot of agricultural problems.
Yes, the similar question was asked by Dr. Raghu. I think, what are your opinions when it is debated with GMOs? And I hope you have clarified it clearly now.
Yeah.
Thank you once again. And the next question is, Dr. Vamsi is putting like, how is this technology would be efficient in terms of cost effectiveness for the researchers?
Oh, this is wonderful question. So genome editing is really cost effective. So if you compare with transgenic crop development, so you need to have a huge infrastructure and also time and also chemical investment. But genome editing, the same construct you could use for one gene to next gene, next gene. Only thing you need to change, there's a small RNA, that's the genetic address that I told you, guide RNA. The small RNA portion you need to change. If you want to modify gene 1, this is done. Now you want to go for gene 2 modification. So you need to change the single RNA. So you don't need a lot of new investment for new modification. So it is highly cost effective. And that is why research poor countries are also using genome editing in a huge amount. So this is highly cost effective, yes.
Yeah, thank you, sir. And the next question is, hybrid crops and genome edited crops is same or different? It is by Saurav Paul.
Hybrid crops, yeah, hybrid technology is different, obviously, but for hybrid technology and genome edited crops, both are distinct. There is no similarities in technological perspective, but for hybrid development, suppose you want to go for hybrid cultivation. Now farmers cannot save hybrid rice or hybrid seeds for the next generation, because if they save the seed for next generation and they grow that hybrid, then the trait is segregated out and they would not get a good yield. So that's the problem. So farmer has to buy seeds from company. Now this genome editing is helping hybrid technology. How? There are 2 papers in Nature Biotech and one is in Nature. So they demonstrated that using genome editing, hybrid technology could be applied to develop some kind of hybrid seed that could be saved by the farmer. So they can save their own hybrid seed for growing in the next generation. They don't have to go to the company to buy each year the seeds from the company. So it is also a very fascinating area that I have not discussed yet. Yeah.
Thank you, sir. The next one is, what is the difference and similarities in Transgenics and CRISPR-Cas?
Can you repeat it, please?
What is the difference in similarities in cisgenics and CRISPR-Cas?
Cisgenics. Okay. So, as I told you, transgenic means you take gene from other plants and you put it in rice plant. That is called transgenic. Means cisgenic means, suppose I know in my PhD work, I worked with a gene that is rice endogenous gene, but that does not express well, means that does not produce a lot of protein. But I know this gene is important for pathogen resistance. So what I did, I isolated rice gene from rice itself. Then I put it back in the rice itself again to produce more protein. Have you got my point? So more copy. And then this crop is called cisgen because the origin of the gene that is you are transferring is from the same plant, same species. That is called cisgen. Genome editing is not like that. Genome editing, you are not inputting any gene from other sources or from rice sources. You are modifying the existing gene, modifying the sequence of the gene. You are not putting any other gene.
Yeah. And the next question is by Dr. Archana Bharati, is that what are the potential risks of agriculture in gene editing?
Potential risks of genome editing in agriculture.
In agriculture, yeah.
In agriculture, we have a very little risk with genome editing, I could say, as compared to human therapeutics. Because, some people say that this CRISPR-Cas9 could generate off-target modification. Now, suppose you targeted gene A, but by mistake, it could also modify in a very little amount, but also modify another gene that you have not targeted. So that's called off-target mutation. So that is a little bit of concern, but for agriculture, it is not at all a concern because off-target mutation could be segregated out in the next generation. So if you grow the plant for next generation, and you could select only those plants that has the only single targeted mutation, but not the off-target mutation. So those plants, suppose you got 10 plants, and out of 10 plants, you get 3 plants that has only single targeted mutation, and 7 plants has, suppose, off-target mutation. So you discard the 7 plants, and you grow the 3 plants for next generation. So this is how you could get rid of that problem.
Okay,
but for human therapeutics, it is very much a serious issue.
Yeah. Moving to the next question, sir. What is the present scenario of CRISPR-Cas in India regarding commercialization?
Okay, that's also a very interesting question. So DBT, Department of Biotechnology, Ministry of Environment and Forest, DBT has already drafted a draft regulatory rule of genome edited crops, and that says that it would not be regulated as transgenic crops, and it would be go freely to the farmers' field without any rigorous regulation as it did with the transgenic plants, because the final crop, the final product does not contain any foreign gene inserted. That is why it would not be regulated. If the changes are few nucleotide change is present in the final product, then it would not be regulated.
Yeah.
Actually, it is following the similar kind of regulation that has been imposed in Argentina, Brazil, and USA, and also recently Japan. So a lot of people are going to deregulate genome-edited crops, so they would not be regulated. Yeah.
Okay. And the next question is, Asif is asking why we have not utilized genomics to its full potential in vegetable crops, but the thrust has always been to cereals and pulses.
Oh, okay. So it means, this is all depends. This is some kind of policy issue, because what crop you need more for your population, and that is why funding also comes. So research funding is more to that crop, and there is a lot of information about that crop. A lot of research has already been published. So this is kind of policy decision. It's not. It's from up to down. It's not from down to up. So how the money is coming to which crop, and which crop is being investigated in more amount, and they do more cutting edge research, and more information is available. So that's why they are being utilized in more number. But if vegetables are also getting funded more, and more research is being published, they would also be utilized in a similar manner.
Yeah, actually, there are a lot of questions, but I couldn't ask all of them due to time constraint. The final question I'm going to ask now, sir, by Dr. Zahur Bayo, like he's asking how to target signaling pathway genes if we have multiple genes responsible for synthesis of salicylic acid. Like that, he given an example that in Arabidopsis, ICS2 gene synthesis salicylic acid. So he want to ask that how to target the signal pathway genes.
Targeting signaling pathway gene also a little bit tricky. Tricky, it means not tricky. Tricky in the sense you can mutate them. Suppose SCS2, if you have one copy of SCS2 and versus multiple copy of SCS2, if you have one copy of SCS2, then you could easily target that gene to mutate that. But you have to always look for if SCS2 has any other role to play, not only in salicylic acid. I know SCS2 also play role in ethylene biosynthesis. So hampering signaling pathway genes sometimes may cause some kind of negative phenotypic impact that you do not expect. Suppose you are only looking for one signaling pathway, but that same protein, suppose SCS2 protein, has a role to play to other pathway also. So if you want to downregulate that salicylic acid pathway, but it can also result into the downregulation of other pathways, and that could negatively impact the phenotype. So you have to be very sure before targeting those kind of signaling pathway genes. But it's similar to any other target. Yeah.
Okay. Thank you very much, sir, for answering all the questions in timely manner, and it was really informative to listen from all the concepts of CRISPR-Cas and genome editing from you. And I would like to request Sneha to proceed further. Sneha, please.
Yeah, sure. Thank you very much, Dr. Mullah, for such a flawless progression of event and for illuminating the subject to this extent. Thank you so very much. So for the next event, I would like to call upon Dr. Sujeshri, Chief Operating Officer of Plant Genomia from Indian Agriculture Research Institute, New Delhi, for carrying forth the interview round.
Thank you, Sneha. Sir, first of all, I thank you so much for delivering such an insightful presentation. Amazing facts and articulation of your complete research results and technologies in depth. It is definitely going to help the scientific community. Sir, now we have a few questions to you to know more about your personal and professional life that audience are very curious about. So, sir, my first question is, what has inspired you to pursue a career in research? I mean, how passionate you were in shaping your career, the winning formula, your winning formula, we would like to know.
Oh, yeah. I in my schooling, all these things I never thought about, and I am going to research. But when I started studying plant science in my graduation, Bachelor of Science, then I got very fascinated to the biology. So once every aspect of a living organism is a story. So there is a hero, role player, and it influences others, and the key player and the side player. So biology was always fascinating to me, and then I pursued masters of science in the same subject. And then while I'm doing Master of Science, I did a project on tissue culture. So I learned how to grow plants without soil and without water in an artificial nutrient media. And then that time I also did one workshop on DNA, molecular biology techniques. That was UGC sponsored, and I was lucky that I was selected for that workshop. Was going on in my department. And that time I first saw a DNA can be cut into two pieces by using restriction enzyme, and it could be visualized with UV light. And then in the next session of that workshop, that 2 cut fragment has been ligated again into a single fragment. So I can say, I always tell that this was my turning point to draw my interest towards molecular biology. So it's wonderful. I did not see any protein or any restriction. I just added 1 microliter of tiny water. I saw it in water. Okay, it looks like water, and it digested the DNA. So then I decided that I has to. That actually draws my interest towards molecular biology and obviously tissue culture. So it was very fast. So I did a potato tissue culture in my MSc. So I have seen a very nice potato growing in a vessel. Okay. I took a lot of photos and showed to my parents. So that's the initial point, and then I qualified the CSR NET fellowship. Then I got a financial security out of that fellowship, And then I immediately joined the PhD program, and I always do whatever I do very seriously. If I have very limited time to execute that work, but I want to be perfect at my level. It may not seem perfect to others' level, but my level it should be perfect. Otherwise, I would not go there. So your presentation today is Sunday. So I was very busy. I worked on my presentation until late night yesterday. So yeah, I presented a lot of presentation in these last few months. I could have simply presented the same slides here. So this is not to boast myself. This is to say you that how I took something seriously. Okay, so that's the hard work, and to be some kind of perfect at my level is one of the, you can say, key to my success in everything. Another key I should say, I always understand the basic fundamental of anything. What I want to say, what I want to write, I never write anything without knowing. So suppose I'm writing some literature review, just I copied one sentence, paraphrased it, and write. No, I first understood everything, the basic concept behind it, and then I write, or then I say that. Because I always fear that somebody in front of me can ask me about the basic concept, and I'd be in a very embarrassed position not to say that, not to answer. So that was the fear point that I utilized to understand the basic concept. So if your fundamentals are clear, then it's very easy to answer any kind of complex question, any kind of problem that you face in your research life, even in your personal life also. So that's the.
Yes, sir. Thank you. And so really it will give us a lot of confidence. Sir, my next question is, sir, you are honored with prestigious Fulbright scholarship, which is offered for the international education and cultural exchange between the countries for the passionate scholars. Sir, could you please share with us the eligibility to obtain that fellowship and the criteria you have met to achieve that before you going to the Pennsylvania State University?
Yes. Yeah, that's a real question. I think that would help to.
Yeah, this is the question which is asked by a lot of participants in the chat box as well. As you are from that background, you would have explained.
Yeah, that's a wonderful question, and thank you for choosing that question. It will really help others. So Fulbright has different section. They award fellowship. So starting from masters. So if you are a master's student, you can get Fulbright master's fellowship. If you are a PhD student, if you can go abroad during your PhD, suppose you are doing a PhD in India, you can go abroad for 9 months, up to 9 months, to do some work related to your PhD in a foreign university. And then you come back and then submit your thesis. Okay? So master's fellowship is there, PhD fellowship is there, and then postdoc fellowship that I got. So postdoc fellowship is really good because it gives you 2 years of fellowship, 24 months of fellowship, and then after postdoc there is professional excellence. Professional excellence, suppose if you are is associate professor or maybe professor or a ranked researcher or academician, then you can apply also to the professional excellence fellowship, where you are allowed to go there for up to 6 to 9 months, and you can carry out teaching, you can carry out research there. Also, Fulbright teaching assistantship is there. So suppose you are a language teacher, suppose you know Tamil very well, you know Kannada very well, so you could go to US and teach them Kannada and Bengali and Marathi and Tamil. Okay, so that's called Fulbright language teaching assistantship. So I would recommend USIEF fellowship website to visit. USIEF, United States India Educational Foundation. It has very good well described website. You could obtain any number of information from there. Even if you require any personal answer also, if you could email me, I could supply you with resources. And I would recommend everyone to try for Fulbright. Fulbright is wonderful fellowship if you just avail once. So there is a phrase, you know, once a Fulbrighter, always a Fulbrighter. So if you be once in your life a Fulbrighter, you would see the immense change that would brought this fellowship to your life. So your confidence level would be here at the beginning, and after you come back, your confidence would be here. You get a lot of exposure. You get to open your mind. You get to see how other people work, do the same work in a different way, in a more smarter way, within the same time. Okay, so and you get to know huge people. You know, you get to develop very good network. So it impacts. I think it kind of U-turn in someone's life once they have a Fulbright. Okay.
Yes, sir. Thank you for your guidance in Fulbright Fellowship, sir. My next question is, sir, can you please share your experience on meeting the Nobel Prize laureates, or the inspiration or the lessons that you have learned to achieve excel in your research from them?
I think, you know, so you see, you guys are very talented. It means you have chosen very intelligent question and very relevant questions. Okay. So first thing I need to say that, so when I met Jennifer, so there was a conference, famous conference, that's called Crispr Cas conference, that happens in Cold Spring Harbor Laboratory in New York. You know, Cold Spring Harbor Laboratory actually generated more than 10 Nobel laureates. And had also Watson worked, James Watson. So even I met James Watson during that visit also. So yes, the conference was going on, and then I applied for a travel fellowship because some $1,000 is needed. So I applied for a fellowship that was a competitive fellowship from Pennsylvania State University, and then I also got it. So I was lucky. So I traveled to New York and stayed there for entire duration, and then yes, I was very excited to meet her, Jennifer Doudna, because I didn't know that because that time she was not Nobel laureate, okay, but we expected that she could get Nobel laureate very soon, and she could be a Nobel laureate very soon. So I met her, and then I said, okay, hi, hello, and then I said, we wish you get Nobel very soon. And then she blushed, you know, out of shyness, and she said, I started my career from Cold Spring Harbor Laboratory. Because she was working with the MIT professor for her PhD, and that time her professor could not join for an invited talk at Cold Spring. Okay, and then Jennifer came to deliver that talk as a student. And Jennifer said, I was shivering out of fear because James Watson was sitting in front of her to listen to her lecture. And she said, that was my first lecture to Cold Spring Harbor Laboratory. And now I am organizing this conference. She was the organizer of Crispr Cas conference. So she said to me that story at that time. So that was a brief encounter with her. She was very humble. And then I have another chance to deliver a lecture at her own institute, at Innovative Genomics Institute, IGI, at University of California, Berkeley. So that time I just published the review in Transient Biotech, and then we are successful in demonstrating in RISE some base editing work. So I was invited to deliver a lecture at her institute, IGI, and I delivered a base editing lecture at her institute also. So yeah, that was the interaction.
Yes, sir. It's inspiring anyhow. Sir, my next question is, sir, you are author of 2 internationally reputed CRISPR-Cas volumes of books of Springer. So with that context, I would like to know more about how you have structured a high quality manuscript that is well conceived and well reported with research studies. So sir, we would like to hear from you the tips and tactics that you follow while framing a content for a manuscript in terms of inclusion of appropriate purposes and rationale, or research questions, literature reviewing, theoretical framework, and describing the research methods and claiming about the results to obtain a very high quality writing.
Okay. What I should say, I always, even yesterday also, when I was talking to my student, so I said to her that writing is not an innate capability, or it is not something that someone is born with. So it's the skill that could be developed. Anyone could develop this skill. Now, as you frequently write anything, as frequent you write, as much you learn. One thing. First, when I wrote my first research article during my PhD, so I wrote myself. Nobody helped me, because I was trained by a very good scientist, very well-known scientist, and he knows how to train a student. So he never helped me in writing. Okay, so he said, you have to write. And I printed out 10 papers. That was my first experience, just I'm sharing, okay? But I'm not like that now. So I printed out 10 different relevant papers, and I sat on a table. And first I have gone through to all 10 papers, and I highlighted each of the points in the introduction, and I said, this point is very good. I could bring it this point to my introduction, okay? So I highlighted, and then for each line, for my first paper, each line I spent a lot of time to figure out how to avoid plagiarism, That time it seems, when you are writing your first paper, that time it seems, no, that paper already printed means we have a concept that is the best line. That's why it is printed, okay? So it is our human psychology that whatever printed, our mind thinks that this is the truth. That's why a lot of false and many things, in writing, they spread like anything, because we always treat any printed thing as a best thing, okay? But it's not like that. So what I did, suppose for 3 related papers, and they explained the similar line in different types, and I explained my same statement, but taking ideas from all 3, and I summarized them in a single language, in a single sentence, okay? So you have to get writing idea from others when you are new. It's not that any idea comes from the sky, or you are very talented that it always comes to your mind. When you are new, when you are young, you have to sometimes imitate a good person, sometimes imitate a good writer, okay? So that is why I learned writing, and now I became a little bit fluent, but I still learn from very good quality articles from annual reviews, the series of reviews they publish, annual reviews of phytopathology, annual reviews of biology, annual reviews of entomology. Okay, so they write a very good writing. So you write good quality paper, if you read good quality paper, while you are writing, you learn how to figure out how to write good things, anything. But if you see in research paper, I always write the portion which I feel very easy to start. Don't start. The research paper has a lot of sections, okay? So don't start writing a section which is tough to you. Start with a very easy thing. So what? Otherwise, you will not get confidence. Suppose when I do a research paper, I always write fast the material method. Why? Material method is the easiest section. You already done that, okay? And in material method section, you could do paraphrasing, okay? You could cite other papers. I followed this one. I followed this one, okay? So when you complete material method, then you get confidence. Okay, I have completed some portion of my paper. Yes. Now, suppose you started another portion that is very tough resultant discussion, and you struggle to write 5 lines. After 1 week, you get frustrated. No, I cannot do that because but write material method fast, and then I write introduction to introduce the topic, and then put the rationale of your research, why you did this research, and then tell that how your research is fulfilling that gap and what you did in a very small few sentences. So that is the introduction. Material method fast, and then I write introduction, and then I write results, and then last is discussion. Okay, this is all.
Yes. Anyway, thank you for your guidance, sir. Yeah, sir. One of my next question is a technical question. Like, sir, we would like to know your perspective in, does the genome editing it can really revolutionize the molecular system, and still, what are the strategies to be incorporated?
Yeah, can you please repeat that one? So I could not follow.
Hello.
Could you please repeat?
Yes, sir. We would like to know your perspective on does genome editing can really revolutionize the molecular systems, or still, what are the strategies to be required or incorporated?
Strategies means at what level? At researchers level, at governments level, at policymakers level. So if I want to answer this question from government level, government should invest a lot of money on, and they should encourage researchers to take out this kind of project. That should fund. Otherwise, we will be far behind from other countries. If you see the recent scenario of different countries employing this technology for agriculture and human health, we are far behind. In comparison to our neighbor. Okay, but we are developed in other aspects, but we are we are far behind in this aspect to utilize the potential of this technology. So the government and other policymakers and whoever in that decision-making position, they have to always encourage any student just come with that kind of proposal. So encourage to take up this kind of proposal.
Okay, sir. Anyhow, sir, that was the last question. And thank you so much for this interesting interactive session. And we are very grateful for your effort that you have taken to share your knowledge and experience with us, sir. And also, sir, we request your kind future support in our scientific forum for enabling us to concentrate more on achieving the various scientific goals. Thank you, sir.
Thank you. So I have one question. You know, your website, I looked at graphic design. It was very nice. Who did that? The designing of your website and
Alamuru is the person behind all this, sir. We are the team supporting him.
No, you do yourself the design.
Yeah, sir.
Yes,
we
sir,
have it by our own.
Our own.
Oh, good.
Yeah,
So
I have designed it by own.
Great.
Okay, sir.
Thank you.
Okay, so should I?
Yeah.
Leave?
Sneha?
Yeah, sorry, actually it's Sunday, so little bit of problem. Sorry, truly an amazing interaction between Dr. Sujeshri and Kodubundan sir. So moving forward, now wasn't that a mind-blowing session we had? So thanks a lot, you guys have a brilliant audience. I want to thank the core support team, Mr. Alamaru Krishna Chaitanya, Dr. Sujeshri, Aman Tutlani, Shrimay Bhattacharya, Kondati Sai Sumant, PV Sriraj, Velamuri Anila, and Sai Teja for making the session successful. Thank you all. And reaching to the end of this, I would like to clarify the process of attaining the certification for the same. A feedback and certification link has been shared in the chat box and the YouTube comment box as well. You're all kindly requested to go on to the same, fill it up, register yourselves. Thereafter, within a slot of 1 week, your certificates you'll be receiving. And if there is some error and you do not receive your certificates after a week's time, kindly visit our official website, www.plantgenomia.com. Please note that the link is valid only for 2 hours. Thank you.
Okay, thank you very much. Bye-bye.
Thank you,
Bye,
sis.
sir.
Bye
Thank
once
you.
again. Yeah, Sneha, you can leave. No problem.
Right. Thank you.
Yeah, all can leave the session now.
Okay.